Actually, good morning, good afternoon, good evening, wherever you are in the world. So I would like to just welcome everyone to our workshop this week. My name is Emad Tajkhorshid. You can see my pointer too, Mohammed or somebody.
Yes, I see.
Yeah. Okay, wonderful. So welcome to our workshop. My name is Emad Tajkhorshid. I'm the director of our NIH Center here. Modeling and bioinformatics, and we developed, as you know probably, the programs VND and NAMD and maintain them and make them available for people. And the whole idea of the workshop, as many of you already know, is to make you more comfortable in working with these programs so you can actually go on and do projects by yourself. So we want to break the initial barrier and make it easier for you to get started with these programs. We won't be able to cover everything about these programs in just 1 week, but again, as I said, I hope that we can give you enough basic information, some of the advanced features in these programs, some introduction to enhanced sampling and free energy calculations that you need to know to be able to do reasonable projects with these programs. Some of the advanced modeling tools that are available in these environments, we want to introduce them to you so you know about them and you know what can be done with those tools, and hopefully one day you can apply them to your projects. And again, as I said, the goal is to make you comfortable with the environment that these software tools provide, so you can actually set up a simulation, run it, analyze it, visualize it on your own later down the road. So in the mornings, we have the lectures going over the theory and the concepts. And then in the afternoons, we have the tutorial sessions, which I heavily recommend. You can go through the examples that are provided in the tutorial, or you can actually use your own protein or system or molecule to go through these tutorials and run into troubles and ask questions and interact and troubleshoot those projects. That's really the most important thing probably you're going to take from the workshop. So we would like to have a hands-on experience for you guys, so you walk the path by yourself and then run into problems. And by solving those problems, you're going to feel much more comfortable down the road. So feel free, by the way, it's an online kind of session. Feel free to turn on your cameras. I'm actually more happy to see your faces while I'm talking. And then feel free to interrupt me. If you have any questions, you can actually ask me directly, raise your hand, or whatever you want to do is perfectly fine. So the more interactive, the better. Okay, so all right, so actually, again, by way of introduction, why we are trying to learn molecular dynamics, it has become actually a very important tool in modern biophysics and structural biology. You can look at the number of molecular dynamics papers published since, I don't know, 30 years ago, and now you can see the growth of MD application in biophysics has been almost exponential. And we are very proud. If you look at the red bars, you can see the contribution that we have made to biomolecular simulation. The yellow curve is just any molecular dynamics in any application. The blue bars show actually the application of MD as a technique in life sciences in biomolecular simulations. And the red bars are actually papers that have used NAMD and VMD. And it seems that actually we are contributing to half of the papers in the field. So that's very nice. Looking at the same data in the other direction is actually how many citations we are getting for our NAMD and VMD programs. And if you look, you can see we have an exponential decrease in the time it takes to get one citation. So right now, it takes only 1 hour and 8 minutes to get one more citation to our papers, which is we are very proud of and happy. So by the end of this workshop, we will have, I don't know, about 100 more citations to our papers. So that's very nice. People are really using our program, and many people use it and don't publish. Many people publish and don't cite us for whatever reason. But people who use it and publish paper and cite us, we get actually every 1 hour and 8 minutes. So this is a kind of the distribution of the users that we have for our software. So if we want to count it differently, I would say we have more than 500,000 users around the world. But if we just focus on users who are downloaded multiple versions of VMD, and we know they are common users of the program, so we have more than 100,000 users, 125,000 users around the world. This is the distribution in the US, and some applications, how they use these programs, for what reasons they use our software. And it seems that we have a pretty broad domain in terms of the application of our software. So, and I have some statistics also on the number of users. We have quite a few Nobel laureates who have cited our applications and software, and we are very proud of that. But again, if you just look at those papers that cite us and their impact factor, you can see that actually a lot of them are among high impact factor papers that have used our software. So I guess the point is just to make it clear that really using molecular dynamics, using NAMD and VMD has become really a major tool, and many important papers and important people are using these software packages. So, okay. So as I said, I hope again, I think I'm gonna change my hat. I was speaking as a director of the center up to this point, but now I'm gonna talk about the scientific aspect of the tool a little bit. So I hope it made it clear that molecular dynamics is an important tool in molecular biophysics and modern structural biology. So let's look at a few examples to make a point. I guess every time you are interested in the behavior of the molecules, these tools become highly relevant. So you can see, for example, on the left figure, you can see the details of interaction of lipids with the protein, which has been very difficult actually to get experimentally. We have some recent cryo-EM information giving you some data on these aspects, but otherwise, MD has been really the tool to tell you what happens at an atomic scale, atomic sort of level in terms of interaction of protein side chains and lipids. So on the right, you can see a better example in which actually we would like to look at the motion of the protein and dynamics of the system over time. So this is something that is impossible to get experimentally, because experiments can freeze the system, take a snapshot at a particular state, or maybe multiple states if you have a lucky protein. But then at the end of the day, connecting these states and providing the trajectory, dynamics, behavior of the system is impossible to do experimentally. There is no experimental method that gives you this spatial and temporal resolution that you can gain from computational work. So drug design is another example of where you might be interested in nanoscale dynamics or behavior of the system. So, oh, I'm sorry. So go back. Yeah. So you can see actually when I took the course of pharmacology, we used to say, oh, there is a binding site, there is a drug, usually, you know, indirectly sort of claiming that there is an area, it should look like this because this drug seems to be affecting the protein, that drug doesn't seem to be affecting. So making indirect kind of conclusions or predictions about the binding site of the protein. But these days, here is the crystal structure of a drug, an antidepressant inside the neurotransmitter transporter, which we know from pharmacology is the main site of the action of the drug. So you have all the information on the left side. You can see more details coming from the structure, no molecular dynamics, but you have information available about individual side chains and how they interact with the drug and how you might be optimizing it and making it better. So if you combine this level of structural information with what you can do with MD, free energy calculation, predicting the binding affinity of different compounds here, you can actually make it much, much faster in industry to develop new drugs or to optimize the lead that you obtain from different sources and predict, actually reduce the number of molecules that should be experimentally tested and synthesized and tested to several orders of magnitudes. It's almost actually impossible to do these things without computational work. So here is another example. Actually, I just briefly go through it. So you are looking at the binding of the ADP molecule, which is a ligand, let's say, to a protein, and how the binding site of the protein shown down below is going to react to binding of this ligand. And you can see this again, that we have the structure in the absence of the ligand, but we can produce the structure with the ligand, which is what we are really interested in through molecular dynamic simulation. This is, I think we published this work, when was it? Yeah, 2010. That was probably one of the first examples in which ligand binding spontaneously to binding site was kind of captured in hundreds of nanoseconds of simulations. But these days you can do these things routinely, actually using MD simulation. So we discovered there is a conserved kind of patch of basic side chain, positively charged side chains that induce this process. And this is pretty common in the entire family of mitochondrial carriers in this work. So several years later, actually a year later, 2011, the group of David Shaw, using the Anton supercomputer, the first generation of the computer, they could show actually that you can run microseconds of simulation for an even better example, a ligand that is in solution initially and then sort of comes, this is a GPCR, G protein coupled receptor, and it sort of binds the protein, explores the protein, and then when it finds its binding site, it stays there. And this was done before the crystal structure was even done for the protein with the ligand. And very interestingly, once they did that, they found that the drug is exactly in the position that was predicted by MD simulations. So it is really powerful. It has come a long way. The force fields are much, much better. We can run much longer simulations to capture really relevant phenomena to biology, to structural biology, pharmacology, drug design, and so on and so forth. So one area that, again, molecular dynamics has been applied has some fundamental issues that we are still working on, is to describe the large-scale protein dynamics. So how, for example, this class of machines, these transporters, for example, undergo very large conformational changes that sort of base their function. So they have to do this to be able to go from one state to another one. And in this case, for example, to pump drugs from the inside of the cell to the outside. So they have to undergo these acrobatic motions, and describing actually these and how they are coupled to hydrolysis of ATP or binding of other ions to the protein is the holy grail of the function of these proteins, actually. And this is an area that is very, very hard to do only experimentally. So usually we try to connect the dots that we get from experiment by doing MD simulation and using advanced biasing techniques and free energy calculation that we're going to cover tomorrow, actually, during the lectures. So, so again, this is another example. Let me skip that. Nanotechnology, this is again another example, another area in which MD can really contribute. So what I'm showing on the left is a kind of chip that experimental colleagues of us developed, in which actually they can detect viruses, this was several years ago before the pandemic, but you can imagine it's very timely these days. They run a sample, and the idea is that if you decorate the chip with antibodies fixed on the surface of the chip, then you should be able to detect if there are floating viruses interacting with those antibodies. And this was the model they were working with. So on the left, you can see it's actually a cartoon. So there is no proportionality of the size. Obviously, there is no concept of dynamics and motion in these, how they might interact laterally with each other, how close they are to the surface, and so on and so forth. So this was the model they were working on. And then we developed actually what is shown here, which is really a molecular picture of how individual antibodies are connected through PEG chains to the surface. So you have some PEG chains collapsed on the surface because they are not connected to antibodies. You can change the density of this. And again, we didn't do very long simulation on this for whatever reason, but actually just building the system and observing some of the properties was something we did. And it was very, very sort of inspiring for experimental colleagues to see if there are laterally interacting at a particular density, how they interact, from what orientation, and so on and so forth. So it provides much more powerful models to explore and to hypothesize about what might be happening at the molecular world. Here's another example, again, a gold nanoparticle that is used experimentally. They are decorating this and to connect certain molecules to it. And you can see on the left side, we have an experimental model developed in Kathy Murphy's lab. Very low resolution, 50 nanometer resolution of this object. You essentially just see something spherical or round, I should say. And this is the cartoon model they used to work with before we came in. So we said, okay, we can do much better. So we can generate a nanoparticle that is shown in yellow on the right side. And then we can sort of decorate it with the sulfur atoms that are going to be connected to these PEG molecules here. Then we can actually use this, put it in a spherical, in a box of water molecules, and even add lipids that are supposed to bind it. So you can already see water molecules, how they penetrate even further down below under the PEG moieties. So you can see really interesting details that you never think about if you don't have accurate models of this sort. And of course, in the using additional techniques of modeling, then we added some lipid molecules or surfactants, I should say, and by applying particular forces, then we can create actually a sort of lipid decorated object that is used in reality, not just a black round thing, kind of. So it gives you much more opportunity to test things and change the conditions and so on and so forth. So let me actually skip this. Of course, then phenomena like binding of proteins to the surface of the membrane and why certain lipids are important for this process. These are very difficult to measure experimentally and know exactly what is going on, because usually you cannot crystallize a peripheral protein with the lipids that are loosely bound to it. So you will lose this fluid membrane environment in your X-ray or cryo-EM conditions. So the simulation is the only way to look at the dynamics. That's really angstrom scale, I would say. So you can see water molecules going through the space formed between proteins in a virus, in HIV actually, and how this might be actually used by drugs to penetrate the virus, or actually the dynamics of it. So every time you are interested in really in the microscopic behavior of the system, and you approach it experimentally, you can also in parallel approach it computationally, and MD becomes really powerful complement to the experimental techniques. So, okay, so I'm gonna show this one as the last example and then switch the subject. So here is an example of an ion crossing a protein that we study under application of electric field in MD simulations. So, and we can see that here under the application of electric field, how the ion crosses the membrane through the channel. In this example, it wasn't even clear why this represents an open channel, but only by putting the protein from the crystal structure next to lipids and simulating it, then we realize that look at the lipids shown in blue and red, how deep they penetrate the bilayer, almost to the midpoint of the bilayer from this side and also from the other side, providing actually completing the pathway that is formed by the protein partially. So without the lipids, by just looking at the crystal structure, you cannot even explain that you are looking at the open channel in the PDB of the protein. So simulations can really help completing the story about. Okay, so let me skip this. So now, one problem that we have in simulation, again, of course, it's not a perfect technique, so it complements experiments very nicely. But then obviously we have, despite the time, despite the spatial resolution and time resolutions, temporal resolution that we have a problem with the time scale of this simulation. Usually, because of the time step that we have to take during the classical atomistic MD simulation, 1 femtosecond, 2 femtosecond or so, then we are limited in the time we can cover. So that's a major limitation in MD simulations. So you have to do, for example, I don't know, 1 million time steps calculating all the forces, propagating the positions and so on and so forth. You have to do it 1 million times just to collect enough for a nanosecond of time. So many things in biology, in molecular systems might happen milliseconds, second, minutes, or even longer. So that's why, so the scope of MD simulation is really limited. And there's a lot of work going into this, and that also translates into the limited sampling of the molecular system. So molecules are blind entities. They have no idea what they're doing. They're just randomly wiggling around, and every now and then, by chance, some of these random motions collectively results in what you might consider a global conformational change of the protein. These individual atoms just moving completely unaware of what is happening to the rest of the molecule. So you're just randomly moving. So you have to sample this process many times to be able to maybe observe what might be happening globally, what is meaningful for you as a biochemist, as somebody who wants to explain the protein. So therefore, you need a ton of sampling in this business. And because we are limited in how many steps we can do, because we are limited in the time scale of every time step, then we can't collect enough sampling from the simulation. So there are a ton of enhanced sampling techniques that we will hear about tomorrow. So that's a very important area of advanced MD simulations that you should be aware of and see how you can apply those techniques to your problems. The other limitation of the simulations, atomistic simulations and beyond, that I would mention is the approximations that we have to make to develop the force field that we use in the simulations. So like, I mean, we are describing all these electronic entities, atoms, as single kind of particles with usually with a fixed charge in our simulation. And that's obviously a sort of significant approximation in this business. So those can also result in some inaccurate information and data in your simulation, and you should be aware of that. You should be careful about those things every time you have an important electronic problem. I don't know, conjugated systems or rotation of a double bond or things of that sort. Electron transfer obviously cannot be described in classical simulations. So you should be aware of this approximation and make sure that you are not over-interpreting what you're observing in the simulation. So saying those two major issues, I think anybody who can solve this first problem probably would be the next Nobel laureate in this business, because it's such an important and limiting factor in running MD simulations. So we have done a lot of work on the hardware side and parallelizing multiple runs, theoretically developing kind of models that allow you to sample the same molecular system in parallel in multiple replicas, what we call, to be able to collect information from multiple replicas and apply them to one kind of system. So there is a lot of work on that front to help with this, but that remains to be an important limitation, and we try to make our software as fast as possible so we can go beyond this speed limit and run every time it's faster, and therefore collect more information about the MD simulations. But again, that said, as I emphasized earlier, there is no other technique that gives you at the same time spatial and temporal resolutions that you can get from the simulations, actually. So that's a major advantage for the technique. So something else that we do a lot, and you hear later actually today about, is to make it easier to set up the simulation, to run them, and to analyze and visualize them. And this is something, the subject of the so-called QuickMD that you're going to hear about. You see a sort of little introduction here of it that allows you to interactively go through all the complicated, sometimes, processes or steps that you need to go on to set up your simulation. So that's another thing that we try to do in our software. In VMD, you have access to this, and you will hear about this a lot. The other application of MD is when you want to put together structural assemblies and complex systems. So very simple example is, for example, when you want to place a protein inside a patch of the membrane. Usually you have information about the protein and no information about the lipids surrounding the membrane. So but by using MD, you can place the lipids initially roughly around the protein, and then use MD to nicely pack these additional elements around the protein. So the same concept applies actually when you are dealing with, let's say, with a viral envelope or capsid that has, I don't know, many copies of the same protein, and you have maybe low resolution information from the experiment, from cryo-EM, from NMR, X-ray. And then MD can come to the rescue. Again, you can start with an initial guess, let's say, of the placement of these elements, and then use MD to allow sort of optimal packing based on the force field and physics that we have sort of modeled for these proteins. So that's an area that is MD has used a lot, actually, and continues to be used a lot. So one major development that we did in our group, so somebody raised their hand. Purva?
Yeah, hi, sorry. Yes, I had a question regarding your previous slide. I wanted to know if the packing that you're talking about, do asymmetric things also, does the symmetry of what you're looking at matter at that stage, or can you do that, apply that to asymmetric complexes?
Right, so for this particular example that I'm showing, there is no symmetry applied. So every individual protein in this case is free to move completely independently. And actually the final model that we have here does have completely, well, I mean, somewhat different conformations of individual proteins in it. So that's what you expect. I mean, it's very unlikely for a real system at room temperature that all protein copies have the same exact conformation. But you can apply symmetry if you want to. We usually don't do that when we simulate things.
Okay.
Makes sense?
Thank you.
Yeah, sure. So one thing that we did for this area is really, I mean, many years ago, you can see this goes back almost 15 years ago, kind of, when the cryo-EM was kind of getting better and better in terms of resolution. Still, we had a low resolution problem with the technique. It wasn't able to tell you exactly where the amino acids are fitting in many cases. So one thing that we did was to develop a technique that we call molecular dynamics flexible fitting. So during the technique, as the name suggests, you do fitting of a sort of initial guess of your protein. It could be another state of the protein coming from X-ray structure, for example, at a high resolution. And then you try to fit that using molecular dynamics flexibly as the protein is moving and doing its natural motion, you try to fit that into the cryo-EM density that you get from a low resolution kind of experiment. So that technique has been used actually very frequently to experimental refinement of a structure that has become almost actually a part of the structural refinement procedure in some labs actually. So that's you can see how MD has been used, has become a part of an experimental approach for getting better structures. So here's another example. You can see we can do it even actually blur the density to low resolution, 5 angstrom in this case, and then slowly change that, making it better and better to allow a protein that is initially in a very different conformation to nicely fit into this structure. So that technique, MDFF, is another sort of example of how you can take advantage of the technique of MD simulation for structural biology. So again, in our group, in our center, we have tried to make it even easier for experimentalists to allow them to use this in an interactive fashion. So we have provided a tool, we call it interactive MDFF. Here is your protein that you want to fit into the density that you see in gray. And then we can run its simulation. And then while it's being simulated and it's sort of molten, allowing you to manipulate the structure, we can go in and interactively apply forces to the system. As you can see in this animation, you can see, so we sort of apply a force to an atom, we come in and using our mouse. So that's not my mouse. So we can, this is in the animation, you can see the direction and magnitude of the force also shown by this arrow, and you can bring it close to the density where you want to fit this element in. So this is something that you can do interactively, so it might not be trivial for the program that you're using for this purpose to do that for you, but we have made it clear, sort of clear to easy for people to do it. At the same time, on the right side, you can see the cross correlation between the structure and the density nicely monitored for you and visualized here. So you can see the correlation between the two objects is becoming better and better. So you are fitting better your protein to the density. So this is again something else that we have available, and hopefully some of you can take advantage of it for your project. And as I said, we use this again every time we are putting together many individual elements. Then you have to rely on techniques like MD to allow the optimal packing. Otherwise, it's impossible for these irregularly shaped lipids, for example, and many copies of the proteins in a cell envelope. You can imagine this could be the, I don't know, the envelope of the coronavirus, for example, or any object that you want to put together. You can take advantage of MD to get perfect packing, to avoid holes, and so on and so forth. So we use this. So on the left side, you can see the chromatophore. So that's a photosynthetic organelle in some bacteria, and that is sort of it uses light and uses the proton gradient generated by these redox proteins to channel it to ATPase shown in yellow to produce ATP for you in the cell. But again, so this is again an example of probably the first big object that was put together, combination of modeling and of course MD. Without MD, you cannot put these things together. And here is another example. Again, I'm just showing through the steps that we can do with MD. So you can put a sphere together using techniques like Fibonacci algorithm nicely, and then replace them with individual lipids. That's a procedure that we developed in my lab to do these things. And then in the second step, you can actually represent your protein with coarse grain modeling, kind of. You know, you have the pink objects representing the membrane part and the blue objects representing the the parts outside the membrane, then use this super coarse grain object. In step 3, shown in red here, you can have as many proteins as you need in your system and allow them to nicely partition in this spherical shape. So they are all actually on the surface of the sphere. Those guys in the back are behind the screen, so that's why they look like that. And then once you have this initial positioning of the protein, this is a sort of very simple MD, by the way, then you can take these, convert them actually to atomistic systems nicely using techniques that you can learn about them today. So you can create perfect holes for them and then add the actual atomistic proteins to these holes to have the complete object that you're interested in and come up with something like this that has I don't know, about more than 1,000 proteins and few million lipids. So these things are impossible to do just based on modeling and placement of the objects, and you have to rely on MD in many, many steps of the process. So, okay, so let me skip this too, because I would like to just switch. Any questions for this first introduction kind of lecture? I don't see any hands raised. All? Okay, Xingchen, go ahead.
Hi, I'm just wondering for the MDFF program, do you have like a limit for the resolution of the map that it's using? Like, does it have to like does the map have to like be better than like say 4 angstrom or 3 angstrom, something like that?
No, actually, we don't have a limitation of. I mean, it cannot be very low resolution because then it becomes very hard to fit the atomic resolution protein that you're working with into those densities. It becomes really, really bad. So we haven't worked with, let's say, maps more lower than 6-7 angstrom resolution, I would say. But again, so what we do, if you have good resolutions, as I showed in an example, you can. Sometimes the problem is that good resolution, high resolution maps, that's a limitation that you need to be aware of, are too rugged. You have too many, let's say, low density, high density region, and it's very difficult for the molecular system to overcome the barriers separating those features in your density. So what we do is to intentionally or manually lower the resolution of your density. Let's say from 3 angstrom, we make it look like 5 angstrom to make it more smooth and less rugged. And therefore, then the protein can nicely slide roughly into the density. And once the protein is in there, then you can increase the density back to 4 angstrom, fit the protein even better, and then to go back to 3 angstrom and fit it perfectly there. Does it make sense?
Yes, thank you.
Okay, good. Any other questions? Okay, if not, let me switch to my next kind of introduction. So here I would like to actually, again, go back to this time scale, just introduce a few things that people have done or we have done to sort of deal with this. So as I pointed out, the time scale limitation is a major limitation in MD that translates into limited sampling of the molecular system. So let me hide actually my panel here, hide floating meeting control. Okay. So what we would like to see actually what has been done to battle the time scale. And then again, as I briefly introduced to you, so you might get a crystal structure of the protein that is here, let's say in one place on your energy landscape. But then during a function, this protein might undergo conformational changes, I don't know, every millisecond or every 100 microsecond to another state outward facing to inward facing and back. And during this back and forth transition, it transport materials across the membrane. And this is something that might not come easily out of the simulation, because you need to do a lot of sampling to observe this transition during simulation. So one area that people have worked a lot on is in the area of lipid bilayers. Many of you might be interested in membrane proteins that you usually place in a lipid bilayer and simulate, just to provide the natural environment of the protein, and to be able to look at it inside the lipid and see how lipids might actually even participate in the function. So the lipid bilayers are very common structures in biology. They're perfect for cellular membranes because they close on themselves. They don't need any additional energy to form these structures. This is all happening in water spontaneously, so that's perfect. The problem is actually then, even though the lateral motion of these lipids could be rapid, at least experimentally speaking, the flip-flop motion of these guys are very slow, 9 orders of magnitude slower than the lateral motion. And this essentially never happens. It's very slow. It happens once in several hours for the lifetime of a cell. Or biology, this essentially means that it doesn't happen. Unless you bring an enzyme, a protein that facilitates this motion from one leaflet to another one, this guy doesn't happen. So as far as the lateral motion of lipids are concerned, this is another important motion, and it's important, for example, for mixing of different lipids that you have in a cell. In cells, we usually don't have any pure lipid bilayer. So it's always a mixture of, I don't know, at least 3, 4, usually more kinds of lipids. And so it's important we would like to probe how they move and how this motion might be important for the formation of different lipid structures, vesicles and micelles and so on and so forth. So even though it's considered rapid in experiment, but it's still a slow diffusion, 3 orders of magnitude slower than water, for example, if you compare it to water diffusion constant. So we would like to accelerate this motion if possible. So one thing that has been done, so this is an example from, I think, 15 years ago, looking at a few nanoseconds, 5 nanoseconds of simulation of a lipid bilayer. And you can see while these guys move up and down, you can easily see that they are not exchanging position at all. So this exchange is slow, and we would like to accelerate that if possible, to get the heterogeneity of the membrane, how they interact with the protein, what's the effect of cholesterol, and so on and so forth. So one thing that has been done to address this particular issue, and actually it's now applied to many proteins as well, is to the so-called coarse graining. So I would call it battling the timescale issue, case 1. So let's talk about coarse graining. And I'll very briefly talk about this, just a concept. And every one of these is days of training if you want to get really deeper in it. But the idea is pretty simple. So you represent the molecular system that we do atomistically in most of the things that we discussed today and this week. You represent it differently. So for example, this head group can be represented by one bead. We don't call it an atom because it doesn't exist, this thing in reality. So this the negative moiety of the head group is represented by another bead, the glycerol moiety, another bead, and the tails of your lipids are represented by 3 or 4 beads. So by doing this kind of representation of your atomistic system, then obviously you would reduce the number of particles for which you have to calculate energies and forces every time step. And then another important thing, which is actually more important, that happens is that now the energy, the potential energy that the terms that govern the potential energy of interaction of these beads become much, much softer. So meaning that the motion of these heavy, big beads are going to happen on a much slower time scale. They don't sort of vibrate like hydrogen atoms that are very, very fast particles. So the motion of these atoms, because their mass is larger, and so on and so forth. So the frequency of motion of these particles is much, much smaller than atoms, and therefore you can take longer time steps for simulating this system. So you have less particles, you can take longer time steps, and by the combination of these two, you can actually increase the time that you can cover. Any question, Ramesh? You had a question? Oh, okay, no. Oh, okay.
No.
No, okay. Okay, so by doing this trick, we call it coarse graining. So because you're representing it as coarser objects here, and by doing this trick, you can actually start observing much longer time scale phenomena like formation of vesicle and so on. Purva, you had a question?
Yeah, so what leads you to decide that you can have 3 larger beads for a part of the section and just one for a certain, how do you make that choice?
Yeah, this is somewhat arbitrary if you develop a coarse. There are many coarse grain models in the field, actually. People do it differently. So in this particular case, this is the case for the so-called Martini force field developed by Alan Mark and Ziver-Jan Marink in the Netherlands, and over the last 2 decades, actually. So in this case, roughly they are going after, let's say, 4 heavy atoms in your atomistic representation mapped to 1 coarse grain bead. So that's what they decided to do. And then they tune the parameters for this coarse grain model to optimally represent the molecular system. But again, if you want to start a new coarse grain model yourself, you can start with 5-to-1 mapping, or I don't know, maybe you can put even 5 lipids together as 1 coarse grain lipid, and then all the head groups in 1 bead, all the tails into 2 beads. And so there are different levels of coarse graining. So there is no, I would say, no real physics. There are AI-based kind of approaches. I've seen articles and papers that try to optimize what would be the best way of mapping this from the shape and the connectivity of the molecular system to these guys. But I would say that still it's a pretty much arbitrary decision that you have to make in the beginning. So we usually use established coarse grain models. They have tested a lot to see actually how good they represent the properties of individual atomistic systems, the collective behavior of these, how good it can be captured by these models, and so on and so forth. So we usually use the established models in the field. Okay.
Okay, got it.
Okay. So then actually, if you do that, then you can see, for example, you can see formation of a vesicle here in this movie that I got from Zivertyan. So, and this is, by the way, the time scale is an issue in these coarse grain models because we really don't have ways of monitoring the behavior of these beads experimentally and to calibrate the system to say what is happening here is, I don't know, 10 nanosecond or 10 microsecond or whatever. Oh, I'm sorry, the movie stopped in the middle. So you essentially just see the formation of a vesicle at the end of this movie. So this is still running, but it's not showing. So, but the time scale that is shown here is just the number of time steps in MD simulation, not the actual time steps or time scale of the process, because things are moving on a different scale here. So we have to do some calibration. Say, if you run 44 nanoseconds of simulation using this model, that means 4.4 microseconds, for example. So we have to do this calibration. And when you have the system that is composed of different components, in this case, for example, water and lipids, it becomes difficult to do this calibration, and we have to make a lot of assumptions. So that remains to be a problem in this field. But as we apply it more and more to the processes that we know experimentally, we can actually put a timestamp a better timestamp on these processes. So this method is actually one way of maybe getting longer timescale processes from MD simulations at the cost of losing some details. And again, for the case of lipids, where the majority of interactions are distinction between hydrophobic interactions, the tail area, and hydrophilic and charge interaction in the head group, this has been very successful and very accurately predicting the properties of the systems. When you take it to other systems like proteins, for example, or nucleic acids, the detailed information or detailed interactions that are involved in those molecular systems matter much more compared to lipids. And therefore, then it becomes a little trickier to use these models more successfully. So, in proteins, you have 20 different amino acids. Every one of them has individual properties that are important for a protein, hydrophobicity, hydrophilicity, charge, shape, length. So these are all the details that might not be captured completely for a coarse grain model in a protein. And therefore, for those cases, we have to be careful and apply additional considerations before we apply coarse grain models to those systems. So, okay. So the other thing that I would like to talk about is this can be exemplified by what we call steered molecular dynamics. So, so in many cases, the process, as I pointed out, the blind molecular system is just fluctuating during the thermal energy that is available into the system, left and right, back and forth, just doing its own thing randomly. Every now and then something interesting might happen to the system. So in general, we are not really interested in those random motions and diffusive behavior of the atom, of individual atoms. What we are interested in is to see if the process that we would like to kind of characterize biologically, biochemically, using experiments, how does that happen? So we are interested in watching the system as it undergoes that particular transition from one state to another one, not what happens in that state too much. Again, there is coupling, of course. The whole process that we are interested in is the result of those tiny, tiny motions of individual atoms at the end of the day. They have to collectively resolve that into that. But actually, if we can avoid some of the random fluctuations, and it's an approximation, of course. So yeah, if we can avoid those things in our simulation, we might be better focusing our simulation or computational time on the process that we are interested in. So one way of doing this is that, okay, I have the force field for my molecular system. Things are moving and fluctuating according to the force field, which are there to describe the natural motion of my protein. But then on top of the force field and the potential energy that I define for the system and interactions, I can introduce additional forces pushing the system in a particular direction. Let's say if I am an atom, so I'm moving around completely randomly left and right, you can add a little force on me to bias my fluctuation to my right. This is my right, I think. I don't know if you're seeing it, to this direction. So this additional of the force then actually bias the motion of the molecular system, and then it sort of makes the system to evolve in a particular direction that you're interested in. For example, me going outside the door, or an ion going through a channel, or two parts of the protein separating from each other, or rotation of the bottom of a rotary machine that you would like to study. So again, this is a non-equilibrium treatment of the system because you are not on equilibrium, and that's a big no-no in thermodynamics, because you describe everything according to thermodynamic properties of the system. Now you are pushing the system out of the equilibrium because of the force that you are applying to it. But then it allows you at least to sample regions of the energy landscape of the space that you might not even sample if you don't apply these two. If you just run equilibrium simulation, thermodynamically sound and reliable and perfect, then you might not even go close to the door that you want to describe. So that's the trade-off of this approach. So you have to decide, okay, to what degree you're willing to sacrifice thermodynamics to at least sample it or take the system to that point. And maybe once you're there, you can let go of the forces and describe the system in that equilibrium, in that point, using equilibrium simulation. So this is the whole idea of state molecular dynamics. And you can, so one possibility that you have is to really define individual forces to individual atoms. Atom A should be pushed to the right by this amount of force constantly, or according to a protocol that you specify in the MD simulation. This part, atom B, should be pushed in the opposite direction, for example. So you have all these possibilities in NAMD by very simple commands that you can select the group of atoms that you want to push or pull, direction of the force. You can specify using those commands very easily, and then manipulate the molecular system while it's being simulated. So this is very powerful. Again, I repeat, so you have to be careful because you push the system out of equilibrium. There is a lot of hysteresis involved in here, but then at least you can sample regions of a space that might not be sampled otherwise. So going back to thermodynamics, so every time you do work on the system macroscopically, so the work that you apply to the system is equal or larger than the free energy difference that you are interested in. So yes, you go from state I initial to state F final. And during this process, you have work and heat exchange, and those contribute to the internal energy of the system or dissipate sometimes. So what you are interested in is to describe the free energy difference between these two states. This could be the rotation of a bond. This could be the opening of a protein. This could be the transition of an ion from one side of the membrane to the other side. It could be a very slight motion of a single atom in your system. So you would like to be able to describe the, describe the free energy, but then again, because you're applying work to induce this process using SMD or another non-equilibrium technique, then in principle, the work that you are applying to the system, part of this work can be actually dissipated to the environment, and then you might lose it, and your work ends up being always higher than the free energy difference that you're interested in. So in microscopic world, so one thing that can happen is that if you look at the distribution of the work, if you repeat this process many, many times, let's say I do it 5,000 times, and look at how much work I have to do to the system every time I do it, and then plot that work. This is the probability of the work. You have the average work here, obviously at the peak. Then the average of the work, we know from thermodynamics that average is going to be larger than the free energy that we are interested in. This number here, this guy, the red curve. But then if you look here, so you can see there is a little tail here. These are cases in which that the work that you did to the system, these are rare events. I emphasize the rare adjective here. So these are rare events in which you used less work than the free energy change between the two systems. And the reason is that these guys, these rare events, maybe and in a microscopic world, they are already moving there because of the collective motion of the system and the thermal fluctuation of the system. They are already moving in that direction, and therefore you applying your work, the work that you need to apply to the system to take it there is less than the actual free energy difference between the system. So now, taking advantage of this rarity, population or rare population, then we can actually define our kind of probability, the quantity that we look at the average and the probability density. Instead of looking at the work itself, I'm going to look at the exponential of the work, e to the minus w over kt here. And therefore, if you look at that one, it has been shown theoretically nicely with Jarzynski and others before him, actually, that this distribution has a perfect match to the actual free energy. So we call this the Jarzynski's identity. So the exponential of the work, if you average that, formally, it gives you the delta G directly. So it has been proven theoretically, actually experimentally. The group of Bustamante also showed that if you pull a piece of DNA hundreds of times and look at how much work it takes, you can get the free energy difference between folding and unfolding of that DNA within less than a kilocalorie per mole. It's very nice and accurate, but again, the key concept here is that you have to repeat this process many, many times to be able to sample this tail of the distribution of the works that you have and contribute here. So you can see this exponential is dominated because of the negative sign, is dominated by low amounts of work. High amounts of work give you a very high, small number here, whereas a small amount of work are going to dominate this average. And therefore, if you sample this region, you are in good hands and good shape. If you don't sample it here and you only do it a few times, I don't know, 10 times, you mostly are going to be here, and the number that you get from this approximation is going to be much higher than your free energy. You're overestimating the free energy, the barriers separating your basins, and so on and so forth. But anyway, the point is actually there is a formality, there is a formalism allowing you to obtain equilibrium properties from non-equilibrium properties and non-equilibrium simulations, but again, it relies on doing the non-equilibrium simulation or process many, many times. So we are kind of restating the sampling issue. Before we had to run very long simulation, and now we have to run many simulations to accurately describe the process. So this is an example. Again, I think I just want to make a point that again, by doing this SMD technique, you can induce processes that are not possible or don't happen at the time scale of your simulations, and then characterize them. So characterize them energetically. So using this technique, actually, we looked at the permeation of a small molecule water. You have 55 molar of it, a lot of it, and how it goes through a channel, and another sort of large molecule going through another channel. So we know that this large molecule only goes through this large channel. It doesn't go through the small channel, but you can induce that in our simulations. You can actually set up your simulation in a way to look at this process, even though it might not be biologically possible. And then you can try to explain why it's not biologically possible. Where are the barriers preventing it from happening, and so on and so forth. There was a question. Sonal?
Yeah, hi, Professor.
Hi. Go ahead.
I wanted to ask, can you use this steered molecular dynamic simulations to test the mechanical properties of the proteins?
Absolutely, absolutely.
Such as collagen?
Yeah, absolutely. So this is, I think I would say that was the birth of steered MD, because people started to do experiments working with mechanical proteins and, for example, using AFM or other techniques to pull on proteins and see what is happening. You can see the force profile, force time profile, force displacement profile, and things are happening as you unfold the protein. But then there was no idea about what is happening to the protein that gives you force peaks as you're stretching it. And that was when people started, actually, Klaus Schulten in this lab and some people in Europe, they started to do exactly the same thing. So let's add a force to one end of the protein, hold the other end, and just pull them apart and see if we can see in the simulation what is happening, what hydrogen bonds are breaking during the process to produce these kind of profiles that we get experimentally. So that remains to be a common area of applying SMD to biomolecular systems. And I believe that our next speaker who is an expert in this area, Dr. Bernardi, might actually show you some example in the next session after the break.
Okay, thank you.
Sure. Yeah, absolutely. Okay, so then, yes, we can induce processes that we know don't happen biologically and then gain some information. So in this case, we apply the force to the center of mass of this substrate, glycerol, through a channel. And then while you do that, you can apply a constant force if you have an idea what the force should be. So let's say 250 piconewton in this particular example. So you can specify that in NAMD. And then apply that constantly. This guy always feels 100, 250 piconewton regardless of where it is. And the direction is always southwest in this example. Or you can do it constant velocity, what we call constant velocity. And I would like to call it variable force. This is constant force, this is variable force. And the beauty of this kind of application is that you don't apply a constant high force probably to your molecular system. You keep increasing the force to the object until it until it starts to cooperate, let's say. And once it starts cooperate, then you reduce the force on it. The way it works is, in practice, it's like connecting a spring to the center of mass of this object, and it starts stretching that spring. So by stretching the spring at a particular velocity, that's why we call it constant velocity, 30 angstrom per nanosecond. By applying that, then you can increase the force, F equal minus kx, and then by increasing the force that is felt by the molecular system builds up more until it has enough force to cross the first barrier. So once it crosses the first barrier and some displacement, then your spring becomes smaller again, and the force that continues to be applied to the system goes down, and you continue to do the same thing again and again and again to describe what is happening to the system. So this is the most common mode of applying forces to the system that we use in the lab. The velocity or the speed that we have to specify here is usually determined by how long you can run the simulation and how long the process that you're interested in. For example, this guy has to travel about 25 angstrom, and then I can run, let's say, 1 nanosecond simulation. So therefore, I specify about 30 angstrom per nanosecond speed to make sure that it completes its transition from the initial state to the final state that I'm interested in. And that's how I put the number 30 angstrom per nanosecond in my NAMD configuration part. So this is again similar to the simulation, to the equilibrium simulation itself. The longer, the slower, the better, of course. But then you are limited by your computational resources, and you have to decide how you're going to distribute or invest your computational resources on the problem. So that's yes, Venkata. I cannot hear you. Venkata, did you raise your hand? Question? Okay, no, maybe no question. Okay, so by doing that, let's say in this case, then we apply the force and we can even qualitatively describe the molecular system. You can see you under the same force, for example, constant force, you can see it spends more time in certain areas because of the interactions being stabilizing the molecular system. You can describe these as binding sites if you wish. There are the hydrogen bonds between the substrate and the channel. And then by applying that Jarzynski's identity that I described, here you can even try to deduce the free energy profile, or PMF, potential of mean force, for the process that you are trying to describe based on the assembly trajectories here. So this is the process. It has been published, so I don't want to spend time on it. But one thing that I would say is that every time you do free energy calculation, this is something that you should keep in mind. And it's commonly used. So your free energy is a state variable. So it doesn't matter how you arrive at that state. So the free energy should be the same. So you can describe, let's say, a sort of long process by many, many segments, and shorter processes, just breaking the entire transition or displacement of the substrate through the channel to individual translocations locally, 1 to 2 angstroms each. And that way you can have actually free energy pieces for each segment, and then you can statistically put them together to get the overall free energy of the whole process. So that's something that we do in umbrella sampling, for example. We describe locally what happens here, what happens here, what happens here, and then we just stitch them together nicely. But this is what we did for this system. And so we pull them here. We start with the crystal structure, we pull it to the left. The same thing, we pull it in another simulation to the right. We do this the same here and here. And then by adding them together, we can get the force. We can get the force for the individual segments, and then the work can be calculated based on that. And by stitching them together, we can get the free energy profile. So in this case, I think we were lucky to get a good number because we had only 4 trajectories, which is by no means adequate for this complicated system. But for whatever reason, we got lucky, and we could describe the motion of the system very nicely, and the energetics of the system very nicely. And you can see that compared to experimental estimates, I would say our number is very nice, 7 kilocalorie per mole, which is even lower than the experimental estimate. But again, the experimental estimates are not very accurate in this case. Delara.
Hello, Dr. Tajkhorshid, do you hear me?
Yes, go ahead.
Okay, thanks a lot for your nice presentation. Can you please go to 2 or 3 previous slides? Not this. Yes, here, please. Here you mentioned that free energy change is most of the time lower than the work. And in rare cases, we see that free energy is near to the work. Yes.
Right.
And we are okay. And we are searching for that rare events. But I want to know how we specify our attention to them and don't consider the other ones. Do you know, because you told that as this function shows, most of the time work is higher, but how we concentrate only on the points that they are near. Thanks a lot.
Sure. So this is, there is no way to concentrate on that, Delara. So this is, as you see, this is a probability distribution. You just have to sample it enough, do it so many times to make sure that hopefully some of your trajectories will capture that rare kind of part of the distribution. You can only do that by increasing the number of samples that you take from this distribution to hopefully to get to that point. There is no way to concentrate on that point, unfortunately. It would be wonderful if we have a way to concentrate that, concentrate our computational effort in that region, that we would be in a very good shape actually. But unfortunately, this can come only, it's like a distribution of, I don't know, people, and you're looking for certain color of eye, let's say. The only way you can ensure that you have sampled that if is that you sample a lot. That's the only approach.
Okay, thank you. So, but my question is that I understood that. So always we have the effect of those common events. So the free energy that we can get is not, you know, because always we have those common events.
Yeah, you have these more common events always, but again, the W is much larger here. And if W is much larger, this negative beta W becomes a much larger negative number. And E to that comes a very small piece in this average.
Okay. Okay, so their effect is lower. Okay,
their effect is much, much lower. So if you have some of these guys, they're going to dominate.
They will dominate. Oh, thanks a lot. That's the point.
Sure.
Thanks.
Of course. There was another question. Somebody else raised their hand. I don't see it now. Okay. Any other questions? Okay. So let me. Oh wow! I have only 14 more minutes. Yeah. Okay. So we can do the free energy calculation. There are tutorials actually in the afternoon sessions that you can, if you're interested in this approach, you can go through and calculate free energies for your own interesting processes. So, 3 people have raised your hands. Can I lower your hand, Delara? Your hand is still, okay. Okay. So, Katerina, question?
Hello. Can I ask you, is it possible to use this SMD to somehow estimate affinity between two protein partners by measuring which force we should apply to pull them apart? Or it's not, there is no this, it's not as...
Yeah, it's not very accurate. Again, I emphasize the importance of doing it as slowly as you can, as many times as you can. For protein-protein interaction, you can maybe get an idea, very, very rough idea, by just separating two proteins, do a mutation, do it again maybe a few times, and then comparing the force profile, the work profiles, and see that in one case it's always high, in the other case it's always lower, two clusters compared to each other. You can do those things, but again, if you are really interested, you have to do it using some enhanced sampling, free energy methods, umbrella sampling at the end. So you can get a quick answer, I would say, but I think these days they are not acceptable, at least for publications, I would say, right?
Yes.
It's a very, very approximate approach to the problem, yeah. If you can combine it with other things, for example, excuse me, if you know the binding affinities experimentally for these proteins, let's say, I know this is, I don't know, 50 nanomolar here, 50 picomolar there, and then I do SMD simulations and I see, actually, yes, I get the sort of the two populations of work that I get are consistent with my experimental numbers. This is very good because then you can go in and see what are the interactions that break and keep the two proteins together and provide a molecular view also for experimentally supported differences between the two proteins, between the two mutations or something like that. But alone by itself, if you have actually, let's say, even if you don't have experimental numbers, you do the SMD, you get two force profiles or work profiles, and you can explain it based on the chemistry. I know what is happening here. Here is the reason. Then it becomes okay, acceptable. It's not very rigorous, but still it's okay. But by itself alone is not showing anything, I would say.
Thank you. I thought mostly maybe it's good, for example, if I have multiple candidates and I don't want to test all of them experimentally, I can top listed them by MD and then bring to the lab.
That's a great idea. Let's say, absolutely. That's actually the best kind of application. If I have, I don't know, 10 candidates, but I can only do 2 experiments because of the limitations in the lab, this is a very good way to screen them and say, oh, these are the 2 most promising ones, and I go after it. Perfect. Yes.
Okay,
thank you.
Thanks.
Sure. Any other questions, guys? Okay, good. Venkata? It tells me that you raise your hand, but I cannot hear you.
Hear me now?
Okay. Maybe there is a problem with your microphone. You might want to take a look.
Hello.
Okay, I want to continue. Okay, so we can get the free energy profile, and in this case, I think if you compare the large substrate and the small substrate, I'm sorry, if you compare the large substrate going through a big protein and the small protein, you can see there is a difference in the free energies. The black one is the big protein, so the substrate does go through it, and there are barriers, so we can characterize the nature of the barriers and where they are located. And then for the small protein, you can see that the entire profile is much higher. I'm sorry, we are looking at the radius, so we can see that the black curve shows the radius profile, the size of the of the smaller protein shown in pink on the left side, whereas the larger protein shows the much wider pore, and that probably explains why the bigger substrates only go through that. And that we captured actually using the, where is my free energy profile? I apologize, I think I missed the free energy profiles. If you compare it, I don't have it on right now. You can clearly see that the effect of the radius is reflected very nicely in the free energy profiles that we artificially created for the substrate going through this narrow pore. So you can see that the nature of the barriers are on the order of 40, 50 kilocalorie per mole, which explains why the big substrate doesn't go through this pore. So the same concept of SMD, so just pulling things, we can also apply it to using the utility of collective variables that we have in NAMD. We can apply it to different types of motions of the molecular system. In this case, you can see we can pull the different parts of the system. We can open angles, we can change the distances between the domains. We can change the radius of gyration of the system. We can change the RMSD of the system with respect to a particular initial point or with respect to a target, if you want. So you can really combine different collective variables. Collective variables are variables, you will hear about them tomorrow much more. But these variables allow you to collectively look at the motion of a group of atoms in a particular way. I already talked about the center of mass. That's a collective variable because it allows you to change a group of atoms, to apply force on a group of atoms at the same time by just specifying the center of mass and what force should be felt by that. You can distribute the forces. So you can do the same thing to a complete helix and look at its rotation. You can look at the angle between two domains of the protein using the same kind of approach. And by doing that, so we have been looking at, excuse me, protein conformational changes in membrane transporters and in other systems, and describe the state transition of proteins as they go from one functional state to another functional state. The same approximations and problems also apply here. Non-equilibrium system, we are probably doing it too fast because it's such a complicated system. This happens on a millisecond time scale. I am doing it in microsecond. So there are the same problems that we discussed briefly for SMD also apply here. But again, it allows you to take the system from one state to another state and see what might happen in the middle. And if you repeat it multiple times, then you can actually get a good idea. And then another good thing about this approach is that it allows you to sample initial, to seed what you might need for better free energy calculation, for example, for umbrella sampling simulations that you will hear about tomorrow. So we have been applying this kind of approach to membrane transporters. This is a family of proteins that undergo these large conformational changes, the so-called alternating access mechanism. They come in different modes and flavors, actually, because they have to switch the accessibility of substrate from one side to the other side of the membrane during their function. And they can only do that by flip-flopping and changing their interdomain orientation and position during the process. So they come in different flavors. These are some systems that we have studied in the lab, and you can see the nature of conformational changes can be different. You can have flip-flop motion, you can have elevator type of translocation of the protein. And this guy is coupled to ATP hydrolysis at the bottom. So they can come really, every protein can play a different music. And by developing this kind of complicated protocol by Professor Moradi, who was a postdoc in the lab back then. So we developed actually this technique to apply the concept of SMD very nicely. We have a tutorial for that as well, by the way, to describe the protein motion and actually get the free energy of the process. The whole idea is the following. Again, we discussed it briefly. So you want to go from one state to another one. And usually in many programs, you can do the so-called targeted MD, in which you just target the target state or orient toward the target state, and you pull the system, for example, using RMSD, root mean squared deviation, toward the target state. Then the red guy gives you the initial guess of the path. And then you can use very sophisticated techniques to refine it to the pink path, which is closed, but sort of minimized optimally or locally. And it gives you a better description of the transition between the two states. But again, if your real path would be complicated and too far from your initial guess, there is no way you can sample that and you can go there. So what we are doing actually is to suggest that, okay, let's do these SMDs using these collective variables that we have in NAMD. You can actually push the protein in many different ways from A to B. And by doing that, by combining distance and quaternions and I don't know, contacts between two proteins, by using all those things as collective variables and your chemical intuition about the system, then you can design SMDs that might actually take you through pathways that are closer to the actual real pathway that you're interested in. And therefore, your initial guess becomes more meaningful, and then you have a better chance by refining those initial guesses, then you can actually get closer to the real pathway. Okay, I'm going to stop here. So any questions about anything that we discussed?
Yeah, hi.
Moher, go ahead.
Hello.
So,
please raise your hand. I'm actually just calling you based on the hands. Moher, go ahead first.
Okay, thank you. So I'm just like curious for the coarse grain modeling, are there like any specific force fields, or can we use the general force fields that for the normal MD?
It really depends on the nature of the molecular system that you're studying. For the biomolecular system for proteins and lipids, I would recommend Martini because it has been used and tested a lot, actually. Okay. They have worked, actually now they have Martini 3, so Martini 2, Martini 3, and they have developed that quite a bit. They have tried it in many, many cases, and very nice description of the process. I know the developers very well, and they are very careful. So I would recommend Martini for lipids and proteins, which is available in GROMACS, actually, unfortunately, not in NAMD. But for other systems, again, especially in material sciences, which was probably even before my biomolecular systems, they had coarse grain models available already there. There could be a variety of coarse grains available. You have to do your homework and find out how good they are and where they fail and so on and so forth. Yeah.
Got it. Yeah. Thank you so much.
Sure. Nitin.
Yeah. So my first question for this regarding this coarse grain molecular simulation only, like, so you were telling about calibrating the molecular system to the time scales. So if you can comment more on that, like if I run something in say Martini, and if I have to relate the time scale, that I can see that my protein is unfolding in say 100 nanoseconds. So is it not that it is particularly 100 nanoseconds? That is what you are saying, right?
Well, I mean, what I said that is there are certain kind of properties of, let's say, standard or reference objects that we can use. For example, let me give you a simple example. Let's say I know what is the diffusion constant of water, right? I know it from experiment, but even if I don't know it from experiment, I can do atomistic simulations and get my force field based on my force field and get my diffusion constant. So I know it's, I don't know, 2 times 10 to the minus 5. And then I do coarse grain modeling and simulation of water, and I see a number there. So by comparing these two numbers, I can find out the time scale that I simulated using coarse grain corresponds to the X time scale in the atomistic simulation by comparing the diffusion constants, right? You understand?
Yeah. Yes.
Okay, so that I can do for, let's say, for pure material, I can do that. So I can then maybe use a different solvent or use a different protein in water. Now that I know what it is for water, I can throw in a protein, doing it atomistically and doing it coarse grain and try to compare it. The problem is that the relationship between, let's say, diffusion constant or time scale of water between atomistic and coarse grain models does not necessarily correspond to the relationship that you have for the protein between the two models. Maybe water becomes, I don't know, 20 times faster when you coarse grain it, but protein becomes 10 times faster only. So there is a disparity between the way you are accelerating these material by coarse graining. You know, because the nature of coarse graining is not exactly the same. You just roughly take 4 heavy atoms of a protein and make it a bead, 4 water molecules making a bead. It doesn't mean that they are now moving, each of them is moving 20 times faster. So that's actually a problem there. So that's something that, again, in order to make it a better guess about the time scale of coarse graining, people refer to these kind of reference states and reference simulations, atomistically or even experiment, and try to kind of match them a little bit, or bring them closer, I should say, because again, you cannot match everything usually in coarse grain simulations.
Okay. So there is no like every time I have to do a test experiment kind of thing, right?
There is no golden number that you can say, oh, this is always 150 times faster. No, there is no number something like that. There are rough estimates in the field. They say, oh, roughly this should be, I don't know, between 50 to, I don't know, 400 times faster, roughly. But again, for everything, if you want to sort of better, it's more like the sort of the seeing the process happening rather than putting some kinetic parameters and properties to it. So that's really hard, actually. So they are not good for kinetic.
No, like sometimes when I read some papers about protein unfolding, what people do is they coarse grain the protein, and when it starts unfolding, so then they start like move to the atomistic simulations. They move to atomic model, and then they like study like what was the triggering factor for unfolding.
Yeah, I would say again, for those, it's nice if you, it's a nice kind of addition to the discussion of the paper to do a comparison of my coarse grain protein unfolding to what is known experimentally or what is known atomistically. It's a nice thing to compare. You should do that, but I wouldn't get my unfolding rates from a coarse grain simulation. Even from atomistic, you have to be very careful, actually, when you do these things. Delara, you have another question?
Yes, please, if you have time.
Yeah, go ahead. I can do one more question.
Okay, thank you.
Another meeting. Go ahead.
Okay, thanks a lot. I have done simulations with GROMACS, and as you mentioned, GROMACS is compatible for the protein and the nucleic acids and lipids. I wanted to know for which system, for which systems the NAMD is preferred. If we want to compare, for example, if we have a system and we want to select which simulation package for that, do you have a comment on this? Thanks a lot.
Yeah. So, sure. So for coarse graining, GROMACS coarse graining is good. So in NAMD, unfortunately, we don't have the Martini 2 and 3 versions available. So if you want to do coarse graining, definitely GROMACS. Now, NAMD, for what system is it better? For what system it's not better? So NAMD uses charm force field, the charm force field. And charm force field is pretty well developed for lipids and proteins, also nucleic acids available, but maybe for nucleic acids, the amber force field might be sort of more readily available and better optimized, I would say. So NAMD is an engine to run the simulation, to do the
Good morning.
But what you're asking is actually what force field is better for what molecular system. So you can do charm in NAMD. That's what usually people use, and that's pretty good for lipids. I would say it's very good. For proteins is also pretty fine after the corrections that they added to the charm force field. For nucleic acids, I have a slight preference for amber force field that you can also use in NAMD or in Amber. Amber has its own MD engine that you can use. So you can use either one, I would say. But for coarse graining, I recommend GROMACS. Yeah.
Thanks a lot.
Okay, sure. Okay, so so I have to stop here. So